Assalamualaikum. We are from SSCG3803, Section 01. We will be presenting our chosen protein with 3D models of dehydrogenase using PyMOS. This is our team members, 6 members in total. First of all, let's get to know the functions of dehydrogenase. So, dehydrogenase is used as enzymes that catalyze oxidation reduction in cells, often using NAD plus or NADH or NADP plus or NADPH as cofactors. They are critical for metabolic processes like glycolysis and citric acid cycle.
Next, I will explain about the reason why we choose PyMol for the molecular structure analysis. The first one is due to the visual capability. So PyMol excel at providing a high quality 3D visualization of a molecular structure. So this is very essential for studying protein configuration and interaction. So the detail and clear representation will help in the understanding complex molecular dynamic. The second one is due to the accessibility. So PyMol is a widely adopted tool in both academic and commercial setting. So there is an ample documentation that was available for users, making it easier for to us to learn and master. The last one, due to the functionality. So the PyMol will allow a seamless manipulation of a molecular model. So there is a features including zooming, rotating, and selecting specific protein for the in-depth analysis. That's all from me. Thank you.
Okay, next I will demonstrate how to use PMOL. Basically, there are three main steps that we use in PMOL. The first one, you need to install and open the PMOL. You guys can download from the website and then click download now. After that, you can choose type of software that we use in the laptop, either Windows or Mac iOS. For the second step, load the protein data, which is to analyze dehydrogenase. You guys need a protein data bank file, which is can find this in RCSB PDB. For example, we need to search dehydrogenase 1BT2. After search the term, you guys can download file and then choose in PDB format. The last step, visualize the protein, which is it will appear like this, and then you guys can click file and then open. Choose the file that we download in PDB, which is dehydrogenase 1BT2. So in slide, it will show dehydrogenase 1BT2 structure in cartoon representation and different color for each. You guys can use all button in left side and also click display on top and then click sequence to explore more about dehydrogenase 1BT2 structure. That's all from me. Thank you.
Next, we will try to predict the molecular weight, the isoelectric field, and the stability of the protein 1BT2. So to predict this, we will be using the tool that is XPETC param tool. We just enter the code 1BT2 of our protein and then it will run and it will show the molecular weights of the protein to be 55,011.28 and it have the PI 5.78. The stability show that the instability index that computed to be 43.66 that classify the protein 1BT2 to be unstable, and the number of the amino acid for this protein is 496. And then we will try to predict the domain of 1BT2. To predict the domain 1BT2, we will be using the PyMol. The dehydrogenase typically has NADH, NAD binding domains that you can use domain database like pfarms for a detailed domain information. From PyMol, we managed to find 3 domains of the dehydrogenase, that is starsuccinase copper-binding domain, polyphenol oxidase central domain, and the polyphenol oxidase C-terminal.
Dehydrogenase enzymes indeed possess unique sites crucial for their catalytic activity and specificity, including ligand binding sites and other functional regions pivotal for their function. It's important to note that ligand binding sites encompass various types of ligands, including substrates, cofactors, and inhibitors. These unique sites play essential roles in substrate recognition, catalysis, and interaction with cofactors and inhibitors. The presence of unique sites can be known through several forms. Firstly, through databases such as the Protein Data Bank, PDB, and UniProt, provide access to experimentally determined structures of proteins and enzymes. Annotation within these databases may include information about active sites, ligand binding sites, and other functional regions identified through structural analysis. Other than that, it can also be identified through molecular visualization software such as PyMol that we chose in this analysis, which allows us to analyze ligand-protein interactions. This can be done by initially highlighting the ligands and amino acid residues surrounding the ligands. These residues constitute ligand binding sites and play a crucial role in interacting with the ligand through various types of interaction. Then, we utilize tools such as surface representation to visualize the unique sites. From this figure, the binding site is denoted by gray. The ligands, light red color within the binding site do not prominently stand out in the surface representations due to its small size and are completely buried inside the protein structure.